So we are going to start with our third speaker. This is Wolfgang Stein from Illinois State University. And the talk is a comparative approach to studying mechanism of neuronal acclimation and adaptation to changing ecosystems.
Yeah, thank you. That was, I love that. This was like almost staged, actually. So I want to start out by saying, that I don't need to convince anyone in this room. I think that the greenhouse effect warms land and waterways. However, I want to make the point that the average temperature that the planet currently experiences is not a problem for animals in their habitat. It's actually the heat waves that become more common with more short-term, rapidly occurring temperature extremes. Right? And you can see this actually on land and in the oceans. And so if on the left up there, you see the number of hot days in the United States in the last 110 years or so. And you can see that they dramatically increased in the last 20 years or so. And on the bottom, this is from NOAA, who might be around for a little longer, comparing ocean heat waves from 1991 to 2024. And everything that's yellow there is a heat wave. Right? So those are clearly increasing. And with that, we have more temperature extremes that are short-lasting but rapidly onsetting. And so what animals are particularly affected by this? I would argue that it's mostly aquatic poikilothermic animals. Most aquatic animals are, with some exceptions, of course. The reason for this is because the heat conduction and convection in water is actually much, much higher than in air. And so these animals cannot seek out shade easily, for example, to cool themselves down because of that high convection and conduction in the water. And so that's a problem. So acute temperature changes are a particular challenge for the nervous system. Neuronal excitability and synaptic activity depend dramatically on temperature. We've seen this now a couple of times today. Changes in excitability and synaptic activity alter neuronal responses and ultimately behavior. So that, thinking about how this works, at the core, neuronal activity comes from the ion channels that are in these neurons, right? May that be the voltage-gated ion channels or the synaptic ion channels. And at its core, we have these ion channels that provide the excitation and the inhibition that make those neurons function. And I'm going to show you an example of a rhythmically active neuron that has a succession alternation between, inhibitory channels and excitatory channels that make that neuron rhythmically active. Could be, for example, a persistent sodium channel, right? Now, if you now expose this neuron to acute temperature change, you heat it up, that alters the, oops, that was not where I wanted to go. What just happened? This, not where it should go. There we go. So if you expose this neuron now to an acute heat or so, this changes the balance of ionic conductances. And in the simplest case, you might end up with too much inhibition, which would silence that neuron. And in the other extreme, you would have too much excitation. You would become hyper excited. And both of these are, of course, pathological, right? So that said, there are some species that do actually quite well despite those temperature changes, right? They expand their habitat range despite these currently occurring heat waves. And so that suggests that their nervous system functions despite all of these thermal challenges. And so that leads us to a couple of really interesting questions. So what are the mechanisms of this temperature robustness? How are these mechanisms regulated? How fast do these mechanisms act, right? Can temperature changes or responses change during the lifetime of an animal? Does robustness differ between species? And since we are talking about species, what species should we actually study? And so I'm going to make a pitch for studying decapod crustaceans here because they have about 15,000 species, about 9,000 live in the oceans. And, we're thinking of lobsters, crayfish, crabs here, right? They live from the cold ocean habitats to shallow, hot intertidals, even freshwater crayfish, right? Or the mountainous areas where you sometimes find those crabs on land. And so they have a high invasive potential that suggests a high adaptability of their nervous system. They are also key species in several ecosystems, meaning they're contributing significantly to the food chain in those habitats. And finally, they have also a high economic value because humans eat them too. Importantly, the nervous system of some of these species is quite well characterized, and I would like to remind you of the stomatogastric nervous system that Eve Marder has introduced us to this morning, so that makes it easy for me. It produces this rhythmic activity that is generated throughout the lifetime of the animal, contributes to the digestion of food, and is vital to the animal's survival here. But more importantly for us as neurophysiologists, is they have 26 really large neurons We have the connectome available. It's shown on the right side here if you care. But more importantly, all of these neurons are individually identifiable. What does this mean? You can take, for example, that green neuron over there, and you can find it, you can find it in an animal, you can record from it intracellularly, membrane potentials, ion channels, and so forth. You can record those rhythmic activity patterns, and you can do this in each animal of the species. You can find the very same neuron. But not just that. You can actually find the same neuron in a lot of different species across evolutionary distances. And I'm just going to show you a couple of those motor patterns from different animals, from different species here, all the way up from the Cancer borealis, which is the species that we have seen this morning. You can easily identify those motor patterns, that they look the same and that they actually have the same neurons, despite the fact that these animals are separated by several 100 million years of evolution. So, going back to those questions, I think decapod crustaceans would be a great species or a great clade to study here. So let me get to the robustness part. Does this differ between species? And so I want to show you the results of some studies that we have done across a couple of different species. So those are 5 species that we've used. You don't have to remember the names, don't worry, but you should remember that we have 2 species that live in colder temperatures, fairly temperature stable habitats. Those are the 2 on the top left. And then we have 2 species that live in temperature variable habitats from very cold to very warm temperatures, and then 1 species down here that lives in a temperature variable habitat but fairly warm temperatures. And so this leads me to a simple hypothesis that we would assume is true, that species that live in temperature variable habitats have particularly temperature robust neuronal responses. And so how do we test this? We dissect the nervous system, we put it in a petri dish. If you can't see it, there it is. It's quite small in some of these graph, like a centimeter or so. We put this under the microscope. You can actually see the cell bodies of those 26 neurons up there. We put it in an EFIS setup, and then we can finally record those rhythmic motor patterns, and then we do the actual experiment where we expose these motor patterns or the circuits to a temperature challenge between around 0 degrees and 40 degrees Celsius to see where these rhythms are active. And so what happens when you do one of these recordings, this is from one of these species that lives in a temperature variable habitat. You can easily recognize the motor pattern up there. You see actually 2 recordings. The top recording is from the pacemaker neurons in this graph. And on the left side, you see a representation of the structure of this rhythm in form of a phase plot. That was done at 10 degrees Celsius. Now, if you go ahead and cool this down to like 5 degrees Celsius, you can still recognize the patterns very easily, right? It's still there, so it nicely functions. You can warm this up to 21 degrees Celsius. The motor pattern is still there, looks still very similar. You can even go higher. You can go to like 27 degrees Celsius. You can still see that motor pattern. The structure has changed a little bit. For today's talk, I don't want to go too much into the details that changed, but what I want you to recognize here is there was actually a difference that I've hidden in there, and that is the speed of that motor pattern. And so when you look closely, you will see that it gets slower at colder temperatures, not surprising, but it also got slower at warmer temperatures. So there is the fastest part is somewhere in the middle at around 20 degrees Celsius or so. And so we found that interesting. So we looked across our 5 species, and what you see here is recordings at 10 degrees Celsius, and then at the bottom at 20 degrees Celsius across those 5 species. And we then compared the speed of that rhythm across these species. And what we found is something quite interesting. On the y-axis, you see the speed of the rhythm, so the cycle frequency. On the x-axis is the temperature. And what you see here is that the motor patterns from animals that live in temperature-stable habitats, those are the 2 top ones, are always faster or better responding to the temperature with a steeper rate than the motor patterns from temperature variable habitats. And so those are the ones at the bottom. And so this was very surprising to us. Eve alluded a little bit to this, that typically we see an increase in the activity or in the speed of these rhythms, not in the ones that live in temperature variable habitats. And we think that might be related actually to an energy problem, or other way around. These animals save energy by having 2 to 3 times less energy requirements to produce all these action potentials while they live in these temperature variable habitats. So that might give them an advantage. However, what happens if we now increase that temperature even more? Well, that gets us to these crashes that Eve has described this morning. So here's a recording at 25 degrees Celsius, where it's rhythmic. At 37 degrees Celsius, this rhythmicity has gone, so this has the typical crash. Not a permanent crash. You can cool this down again. It comes back. But what does this look like across those 5 different species? And so at the bottom now, you see that all of them do crash eventually, And if you look at the temperatures, they're quite different. And so let's take a look at what this looks like when you do a quantitative comparison. And so you see the crash temperatures plotted here. On the left side, you see the crash temperatures for the temperature stable habitat animals. Those are the ones on the left, right? And you see that they are significantly lower crash temperatures than the ones that live in temperature variable habitats. Okay, so what does this mean? That means that neurons that in crab species that live in temperature variable habitats, first of all, show a very broad temperature robustness, about 30 degrees Celsius or so, which is very high. And neurons in crab species that live in temperature stable habitats show a smaller temperature robustness and potentially energetically more costly activity. So that supports our hypothesis that I mentioned earlier, that species that live in temperature variable habitats have particular temperature robust responses. So, going back to our questions, can temperature responses change over time? So what you see here is an analysis of one of these species that lives in very cold and warm water, so temperature variable habitats. What you see is a temperature scale from 0 to 40 degrees on the right here, and each of these horizontal bars is one animal, so you see 10 animals total. When it's green, the rhythm was nice and present, so functioning. When it's yellow, the rhythm became intermittently active, and when it's brown, this is where the rhythm crashed. And what I want you to see here is that there's about a 30-degree range in which the rhythm was functioning and there. So that was nice. That was what I showed you before. But now I'm asking, can this be acclimated? And so what we did is we took the same animals. This was acclimated at 8 degrees Celsius. And we took them and put them into 15 degrees Celsius for about 2 months. Coming back to those for a couple of weeks later. And asked, does this change? And what you see here is what you should immediately is that all these bars shifted to the right. So now instead of like a crash at 33 degrees Celsius, you see a crash at 37 degrees Celsius. So this shifted to the right. And even at the bottom, you see a slight increase in the temperature at the cold crash from 2 degrees to 3 degrees or so, a small change there. So do temperature responses change over time? Yes, they do. Acclimating animals to different habitat temperatures shifted that range of temperature robustness. We actually found similar results also in wild measured populations that I have not shown you today, but that suggests to us that similar mechanisms act also on those populations in their natural habitats. So, how fast do these mechanisms act? And so I can't give you a full answer here because we have only preliminary data, but this comes from one of these species, again, that lives in temperature variable habitats. These were acclimated for 2 months to 10 degrees Celsius. Here is 2 months, 20 degrees Celsius. You can see those bars start to shift to the right. What about a time frame in which heat waves actually happened, like about 2 weeks or so or shorter? This is what is in the middle. We only have an N of 4 of this. This was done by my master's student, Charlotte Steiger, who's in the audience. We don't see a change at this point. So that gets me back to those 4 weeks that I've just heard in the talk before me. So that seems to line up. Okay. So what I've shown you, neuronal temperature robustness correlates with and responds to environmental temperature conditions. Acclimation mechanisms are rather slow. We think weeks rather than days, which questions whether these neuronal responses can actually adjust to rapid heat waves that we currently experience. Okay, so here are my 2 last slides that I was asked to put in. Scientific gaps, unmet needs, and so forth. The mechanism of acclimation I clearly haven't put on there. So how do we have to think about this? Well, it will ultimately come down to these ionic conductances in these neurons. And so we have to think about the same lines as ionic conductance that support the acute temperature robustness when we think about acclimation as well. And so that would be like the regulation of ionic conductances. For example, a persistent sodium channel could be there, or an H current or so. But we have to keep in mind that there are degenerate solutions to this, so this is going to become very complicated to study. We think about neuromodulation. That is something that I haven't shown you today, but that my lab has focused on quite a bit. Those are neurons that respond to temperature and release neuromodulators that make our circuits more robust. We have to also think about membrane fluidity, because that one affects ionic conductance quite dramatic. It's usually not something that, as physiologists we think about a lot, but it does happen. And then, of course, there are probably a lot of other mechanisms that I'm not thinking of right now. We have to also think about where does this happen on the genomic level for if you think about adaptation as opposed to acclimation, transcriptomics, right, or transcript level, protein level. We've heard, RNA editing before today. ultimately, physiology and behavior is the output, right? We also have to think about developmental aspects of this. I have some colleagues who work on this in Germany that collaborate with us on our project here. None of their data is currently in this talk, but hopefully next time I can present a little more about this. And then finally, outlook, right, for the future of neurobiology in changing ecosystems. Acclimation versus adaptation, I think we need to think about whether those mechanisms are the same or distinct. Can we find the same mechanism actually? We need more mechanistic investigations instead of just observational ones, right? Common garden experiments, controlled experiments in natural habitats or in simulated natural habitats, right? Which the ADC probably will get at. We need to think about, those mechanisms across clades. And what we need for this is shared tools and ways to make their data accessible, and then, of course, collaborations across our little islets of research. With that, I'd like to thank my lab and the people who are involved, in particular my collaborators in Germany as well, and then, of course, the CAFLI Foundation for their generous funding. Thank you.
Thank you for the talk. Do we have a question? Yeah.
Really cool work. My name is Matthew, by the way. So you kind of touched on this, but is there a trade-off for robustness? And I mean, you could kind of think that being robust could allow like a crab to inhabit different, colonize new environments and things like that. But once it establishes itself in a more stable environment, why would it need to then, de-robust anyway?
Yeah, I think there's cost to it. Part of this might actually be in the idea of degeneracy. And someone asked this earlier today, is there a change in the solution space as you acclimate? And I think there is. So you might, as a species, lose out by having less variability and less inter-animal variability as you acclimate to different habitats, particularly if it gets warmer. And so there might be a cost in terms of the species survival to this, but for the individual animals, I think the survivability goes up, but then as a species, there might be costs to this. And there are, of course, metabolic costs that I completely ignore in any of what I do. Yeah.
Sonal?
Hi, I'm Sonal. I'm a postdoc at Brandeis. I was wondering, so when you look at the species that have naturally a wider range versus the ones that have a narrow range and therefore change. So the narrow range, of course, respond much more strongly to temperature as you increase it, and the wide stay stable. Do you think these are differences in the kinds of solutions they apply, or their ion channel proteins might look completely different so that they don't have the same sort of temperature responsiveness? Where do you think those differences might arise from?
Maybe I'm not sure if exactly understood what you meant, but did you say that the cold or less variable habitat ones don't acclimate?
No, I meant the Q10s basically. A flat Q10 versus a steep Q10. And the ones with the narrow range, the stable ones have a sharper Q10, and the ones with the variable have a flat Q10. And I was wondering if you have any ideas about where
okay, like you're thinking about the frequency of the rhythm in particular. Yeah, I don't have a good answer to this, so I'm not sure how the regulation of the cycle frequency changes between those species or why they would not show that steep increase. What we did see, though, is when you get to higher temperatures, their frequency starts to increase. So for most of them, there was one species in there that doesn't, and I don't understand why, because they are essentially flat. But for the ones, for most of them, they start to come up as we get to the extremes, right? And so in sort of like a, very simplistic view, I would say, like if you're getting to like really warm temperatures for the cold, the ones that are used to the cold, they start to increase, and then the ones that are used to the warm, they start to increase also, but at the higher temperature. There might not necessarily be a difference in the proteins or so.
And one last question from Martin.
Hi, great talk. You've been listening to our talks for the past 6 months, so it was nice to listen to your talk for a change, and now I have 15 questions for you. Now, my question is, you mentioned that potential mechanisms are involved channels or membrane fluidity. What do you envision as being like a sensor for triggering these changes, like stretch channels or metabolites secondary to changes in metabolism or?
I have a feeling it's not metabolism, but I don't have any evidence that it is or is not. Of course, this is a good question, and this goes also, at what we've just heard before. My first response would be like, key chalk proteins, TRP channels, Q channels, those kind of things. The answer is we really don't know. That is something that we are very interested in particular because we have also upstream neurons that innervate these circuits that we know release modulators that help with this response. And we know that those are temperature sensitive in a way. They change their activity, like very similar to what we just seen before, actually. But we don't know how they respond to temperature. They don't have any sensory input, so it must be within those neurons somewhere. But I don't have a good answer, sorry.
Thank you so much. An applause.